Уважаемые коллеги, мы специально начали конференцию с доклада психиатра, поскольку она проходит в психиатрическом учреждении, но все-таки в заглавии конференции звучит фармакогенетика, поэтому нам надо балансировать клинические и генетические доклады. И следующий доклад у нас генетический, доклад доктора Донована, генетические исследования шизофрении, современные достижения. Доктор Донован, пожалуйста.
Thank you. I'm not sure what the background of many of you in the audience is. I think maybe there are clinicians who might not know so much about genetics, psychologists, and maybe some nurses, maybe some geneticists. So I'll try and not speak in too much detail about the technology or the sort of methodology, which is really for people who are immersed in the subject. I'm just going to try and tell you about where we are now in the field and what some of the results look like and one or two of the applications. Okay, so we've known for a long time that schizophrenia is partly heritable, and we've known this for many decades. The question is, so what? When I was a young psychiatrist, we had to learn about twin studies. We had to learn about recurrence rates of illness in families, and that used to bore me because I didn't know what I was supposed to do with the information. We would see that if you had an affected identical twin, you had a 50% chance of getting schizophrenia, but that didn't really help you with the understanding of the illness. With the 1980s, the technologies of modern molecular genetics began to become on stream, and instead of just knowing the rate of illness in various classes of relative, it became possible to begin to think about identifying genes. And the whole point of identifying genes, or the main point of identifying genes, is we believe if we can identify the genes, we can identify the proteins that are made by the genes, and if we can identify the proteins, we can begin to figure out what's going wrong in the brain cells. And if we can figure that out, we might be able to understand what's going on at the level of the whole human being and how they work in their environment, what makes people different, and how we can maybe treat them differently with specific treatments, and maybe even prevent the illness. So that's the big hope. In the time that I have, I can only really talk about the genetic bit. We are doing a lot of work in these other areas, but in 20 minutes, only the genetics can be discussed. So, for the first 10, 12 years of my life working in genetics, it was a waste of time. We found nothing. And we found nothing that was reliable and reproducible. And that's because we didn't have either the technology or the sample sizes required to generate robust results. And just over 10 years ago, I coordinated this study, which was the first successful identification of a risk gene for schizophrenia. And in many ways, we only did this by being lucky. So we had what was at the time the world's biggest collection of DNA samples from people with schizophrenia. But really, it was only about 800 samples from about 800 patients. And we made this finding, and then I had to get on the phone and phone friends in the United States in Germany, in China, in Japan, lots of different countries, and persuade them that we had this interesting result, and would they look at it in their own DNA samples and send us the results, and we could put it all together and have one big analysis. And I was successful in persuading people to do this, but ultimately we had to study DNA from 20,000 people with schizophrenia in order to really prove that this was a true finding. So that took several years, and it became clear that if we did this every time we had a finding, we would never find enough about the genetics of schizophrenia if it took several years to identify one thing. And so people became interested in working together in a more formal way. Originally, there were some consortiums. I know that some people from Russia were involved in the S-Gene consortium that was led by Decode Genetics in Iceland. I worked with the International Schizophrenia Consortium, and there was a number of other ones. And between us all, round about 2009, we had made 5 findings, and that was all working together. And that really, we were quite pleased, but it was not enough progress. So instead, the various small groups, consortiums, got together to form the Psychiatric Genomics Consortium, which has now grown and grown and grown bigger and bigger. And what I have plotted here on the bottom, the x-axis, is the number of cases we now have in terms of thousands. And on the other axis, the y-axis, I'm plotting the number of findings that we have. And basically, the more samples we've got, the more we find. For every 1,000 new cases we get, we find another 4 findings. That's roughly the ratio. And so today, what I have plotted here is, as the previous speaker mentioned, a Manhattan plot. On the x-axis, for those of you who aren't used to this, these are all the chromosomes stretched out from chromosome 1 all the way to the X chromosome. And on the y-axis, I have the level of statistical significance. I have a red line. If the dots are above the red line, these are strong findings. And now we have about 70,000 patient DNAs and about 100,000 control DNAs, and we have 330 independent findings spread across the genome. Each one of these has a small effect on disease. The odds ratios are typically they increase the risk, each SNP increases the risk by less than 1%, so very small effects. But there are thousands and thousands of these to be found. So we've found a lot, but we need to find much more. An important development is that up until recently, nearly all the genetics was being done in people of European ancestry, either living in Europe or living in the United States or living in Australia. Recently, with the help of the Psychiatric Genomics Consortium, we encouraged a group of people working in China and Japan, Indonesia, Korea, to develop their own East Asian Schizophrenia Consortium. And now they have a reasonably good sample size of about 25,000 cases, and they too have begun to make findings. The important message that comes from that study is the genetics of schizophrenia in Asia is the same as the genetics of schizophrenia in Europe. This means, they have the same risk alleles, they have the same genetic effect sizes, and what this means is that there aren't really very important environmental gene-environment interactions between Asia and Europe. To the extent that people in Asia have different cultures, are exposed maybe to different drugs of abuse, whatever, they're not interacting specifically with the genetics. So we don't find evidence for the much looked after gene-environment interaction. In terms of rare, so these variants I've been talking about so far are common. There are lots of rare genetic variants. These have been known about for quite some time. What I have pictured here is a deletion on chromosome 22 called, sometimes it's called VCFS, or velocardofacial syndrome. And people who have this bit of chromosome 22 missing have a big increase in risk of schizophrenia, maybe 25-fold increase compared with these small SNP effects. And my colleagues, particularly George Kirov, but others have now identified about 12 of these types of mutation. Each of them makes a strong risk of disease if you're unlucky enough to carry them. But in total, maybe only 2% of people with schizophrenia carry them, whereas for the other ones, everyone carries some. So these are rare, but important. But even these, if you have one of these, it's not enough to cause the disease by itself. You also need some of the common genetic variation. More recently, we've started doing big DNA sequencing studies. So here, the copy number variants that I was talking about before, the big deletions of chromosomes, they often affect maybe 1 million of your DNA nucleotides. What we're looking for now is single changes in the DNA sequence that change a single protein, or they destroy the function of a single protein. So now we're looking for single small mutations, but we're looking for rare ones that have big effects in disease. And the first success here was done by a consortium known as the UK 10K Consortium, which I was a part of. And basically, we found a number of very rare mutations in this gene called SETD1A. The importance of this gene is it will be one of many, but this is this regulates, this is an epigenetic modifier. This changes the chromatin structure of many genes and switches them up or down, so it controls a lot of things that themselves will be relevant to schizophrenia. Most recently, my colleague Elliot Rees in Cardiff, we've been looking for mutations called de novo mutations using DNA sequencing. And here we're looking for a single base change that someone has that neither parent has, so it's been a new mutation. And what he's found is that there is multiple new mutations occur, again rare, in this gene called SLC6A1, which is a master GABA or gamma amino acid reuptake channel. So this tells us that GABA is very important in schizophrenia. And people have had theories about this for a long time. Now we have very strong genetic evidence that just by disrupting GABA transmission, you can increase risk of schizophrenia massively. And so what we would hope is that perhaps by adjusting GABA transmission in a different direction, we can help people with schizophrenia. Now, just as I've been talking about consortiums to do with finding the common weak genetic variation, you also need very big samples for the DNA sequencing. But DNA sequencing is very expensive. And so these are very challenging studies to do. Nevertheless, we are coming together in consortiums to do DNA sequencing. And this is the biggest consortium, is called SCHEMA. And it's led out of the Broad Institute in the United States by Ben Neal and Mark Daly. And we've now sequenced between us about 25,000 cases, and we have data from about 100,000 controls. And what we've now found, we found another 10 genes with these rare, big, penetrant mutations. Again, these genes are not enough on their own to cause schizophrenia. They maybe take you 50% of the way there or 80% of the way there, but you need something else. You need common genetic variation or you need a relevant environment exposure. And what we're beginning to see is a lot of the, or some of the genes that are being affected by the common weak genetic effects are the same genes that are affected by these different type of mutations. I've given 2 examples on this slide. GRIN2A is a glutamate receptor. And so again, there's been a lot of interest in glutamate in schizophrenia for a long time. We now have good genetic evidence for that system. SP4 is another one of these genes that regulates the expression of many other genes. And so people have to try and figure out what that's doing in schizophrenia. So to summarize the genetics, we now have about 300 strong genetic findings in schizophrenia. What I've plotted here on the x-axis is how common each genetic variation is. And we found lots of ones that are common, like in 1%, 10%, 20% of the population. Everyone in this room will have several hundred of these. Everyone. There's no one who doesn't have any of these. What in the case is the people with schizophrenia have more of them, but we've all got them. And then these are a weak effect, and then we've got these other ones that I've plotted, which have got much bigger odds ratios, much bigger effects, but you can see how rare they are. Some of them only occur in 1 person in 100,000, so some of them are extremely rare, but they can be useful for telling us about the biology of the illness. And so in one such, or series of studies that we've published, we took all the genes that map or are located within the common, the copy number variants, or all the genes that were affected by some of the rare de novo mutations, and looked to see what do they have in common. We find lots of genes, but they must be doing something in common. And what we found was that they were mapping, hitting this complex called the postsynaptic density. This is an important regulatory set of proteins, the terminals of glutamate synapses, and these then regulate cognition, memory, learning, all sorts of very important biological functions, including schizophrenia. We can also take, I don't know what the history in Russia is, but when I was being trained, there were lots of people saying, well, schizophrenia isn't an illness at all, it's made up, it's not true. Or schizophrenia isn't a brain disorder, it's a disorder of the bowel or the foot or the smell or whatever. What we can do now, we have, with these 300 or so genetic associations, we can say, well, these genes, where are they active? Are they active in the gut, or are they active in the brain? If they're active in the brain, which type of cell are they active in? And what we've been able to show through a series of studies, on the right-hand side is the most recent data, that really these genes are active in neurons. The signal is mainly neurons, not glia cells, not immune cells, not cells in the stomach or the bowel or whatever, but neurons. We've been further with the most recent data. So in addition, the geneticists need to work closely with proper cell biologists or neurophysiologists or you name it. We've been working closely with this consortium called the SINGO Consortium. And what they've got like 1,000 experts across the world, and they've been putting together the protein complexes and trying to understand which genes are expressed, which proteins are present in which compartments of neurons and synapses. And what we've been able to show in this cartoon here is that the genetic signal is not just particularly in neurons, it's particularly in postsynaptic structures of the neurons, and it's not just any old postsynaptic structures, it's particular specialized bits of membrane in the postsynaptic neurons. So this is something that we are writing up at the moment, and that should be publicly available soon. And so I think I've pretty much used all my time. Just to summarize, the genetic findings are accumulating very fast, but there's much more to be found, and it will only be found by collaboration. The interpretation isn't simple, but the resources to help us interpret it are improving. We've seen rare and common genetic variation converging on the same genes and the same biology. And there are many, many other applications of genetics other than trying to do this biology stuff. Other speakers will be talking about them to do with introduction in animals or doing prediction or many, many, many other areas of science. And with that, thank you for your attention.
Thank you very much, Dr. O'Donovan. We have time for a couple of questions, maybe. Any questions, вопросы, коллеги, к доктору О'Доновану?
Евгений Михайлович, у меня есть не вопрос, а небольшой комментарий. Во-первых, я хотел бы с гордостью сообщить о том, что среди нас есть два участника консорциума PGC. Это Вера Евгеньевна Голембец, следующий докладчик, и Анна Эмировна Гореевна из Уфимского научного центра. И, опять же, это важное достижение для нас. Первый завершенный проект нашего национального консорциума по психиатрической генетике позволил нам стать, да, Рауль Радикович, участниками огромного консорциума PGC, о котором рассказывал профессор О'Донован. Если есть вопрос, один, пожалуйста.